Bifidobacteria are bacteria found in the digestive tract of all humans. They are considered probiotic bacteria, meaning their presence in the body is beneficial. They are adhesive to intestinal epithelial cells, which absorb nutrients and protect against harmful substances. When bifidobacteria adhere to these cells, the gene expression is affected, which is what makes them beneficial to our bodies.
One example of altered gene expression is chemokine activity. Chemokines are signaling proteins that recruit cells from the immune system to a site of infection, which causes inflammation. Bifidobacteria alter the gene expression of intestinal cells so that there is reduced chemokine activity, resulting in less inflammation.
Human milk oligosaccharides are a prebiotic unique to human breast milk that serve as a metabolic substrate. HMOs are indigestible in humans, but their relationship with bifidobacteria in the intestines of infants make them a necessary component in breast milk.
The researchers used KEGO2 cells, which are manufactured cells that highly resemble human intestinal cells. They cultured these cells before beginning the experiments. The researchers wanted to observe two different effects that the food source had on the relationship between the bifidobacteria and the KEGO2 cells. They wanted to first see how the food source affected the adhesion, and second, they wanted to see how the differently fed bifidobacteria affected the gene expression of the KEGO2 cells.
In order to test how the food source affects adhesion of the bifidobacteria, the researchers first fed two strands of bifidobacteria, B. breve and B. infantis, with the three food sources: breast milk, glucose, and lactose. These bacteria were then separated, centrifuged, and incubated into cell monolayers for 2 hours. These monolayers were then washed with PBS. The bacteria that did adhere to the KEGO2 cells were then quantified through quantitative PCR, a process of amplifying DNA with a polymerase chain reaction in order to quantify a particular gene.
The results of this part of the experiment showed that B. infantis adhered to the KEGO2 cells better than the B. breve for all three food sources. In this figure, you can observe that B. breve did not adhere very well to the KEGO2 cells with any of the three food sources. However, B. infantis adhered significantly well when it was fed with HMO. The bacteria did not adhere well at all, at less than 2%. This shows that feeding HMO to B. infantis showed the greatest rate of adhesion to KEGO2 cells.
The second property the researchers wanted to test was how different fed bifidobacteria affected gene expression in the KEGO2 cells. They did this by incubating the B. infantis and B. breve cells with KEGO2 cell monolayers for 2 hours. They then washed these cultures with PBS and performed gene expression analysis. The genes with the most significant changes in expression levels were analyzed further.
RNA sequencing revealed that there was significant downregulation of gene expression of chemokines in KEGO2 cells that were fed with HMO. Chemokines cause inflammation of the intestines, and they play an active role in development of gastrointestinal diseases marked by inflammatory responses such as ulcerative colitis and Crohn's disease. Compared to the bacteria that were fed lactose and glucose, the bacteria fed HMO showed a much higher rate of downregulating genes responsible for chemokine activity, thus reducing inflammation in intestinal cells.
This research provides further evidence that bifidobacteria is extremely important for the immune system of infants. The use of bifidobacteria as a probiotic in order to promote a balanced intestinal flora is a huge application that could change the lives of infants. The taking of bifidobacteria supplements at a young age can help protect adults from inflammatory bowel diseases that are significantly regulated by HMO-grown bifidobacteria. The first infant formula containing bifidobacteria was introduced in 2007. This is mainly used for infants as a supplement, which naturally contains bifidobacteria.
The main application of this research is knowledge regarding the food source that bifidobacteria probiotics must be given. The prebiotics used for bifidobacteria must be similar to HMO in order to enhance its probiotic qualities.
This article shows many applications of material that we have learned in class. In fact, it covers material from all three modules we have discussed so far. It talks about the carbon source of food required for cellular respiration in bifidobacteria. It discusses RNA sequencing and how the food sources affect gene expression. It also discusses evolution by mentioning that there is a cluster of genes on bifidobacteria for encoding milk oligosaccharide metabolism, which suggests the co-evolution of the bacteria with milk.
To summarize everything, this experiment looks at the change in gene expression of inflammatory genes in intestinal epithelial cells evoked by bifidobacteria grown on different carbon sources. The results showed that bifidobacteria adheres to cells best when grown on HMO, and that the bifidobacteria grown on the HMO downregulated the expression of genes related to inflammation. The information uncovered in this experiment can be applied to the use of bifidobacteria as a probiotic to promote gut health and prevent inflammatory bowel diseases. There is also support for HMO as a prebiotic in order to promote the growth of bifidobacteria within the host and inducing protective responses.